Yes, sir.
Yes, the screen is visible, right? This is what we did. Okay, so quickly, let's start with a brief recap of the last class. So who would like to give me a recap of the last class? Quick revision. Let me go to Rifat. Rifat, are you there?
Yes.
Yes. Would you like to please give a quick recap, a quick revision of what we did in the last class?
We discussed about Mendel's experiments.
Yes. Okay. Start.
He experimented on pea plants and he found characters.
Yes. Why did he choose pea plant?
Because it has a short lifespan and many contrasting traits that will be studied,
Yes.
and it has self-pollination and both self-pollination and cross-pollination is possible, and a pea plant produces many progeny.
Right. And then what were the characters that Mendel studied?
Height, seed color, seed shape, and flower color, pod color, pod shape, flower position.
Yes. So there were dominant and recessive traits in these characters, in each of the character, right?
Yes.
Yes. And then what we discussed after that?
And then discuss allele.
We also discussed chromosomes, gene, and alleles, right?
Yes.
Yeah. And what were alleles?
They are the alternate versions of genes, and they giving rise to contrasting traits.
Yes.
And then we discussed law of dominance, which it is present in a heterozygous condition.
What is this law? What does this law states?
It states that one allele is dominant over the other and always expressed in presence of recessive allele.
Always expresses in a heterozygous condition.
Yes.
Yeah. Then we did
dominant trait and recessive trait.
Yes. So you understood how to do crosses for one gene inheritance?
Yes.
Yeah. In the end, I also told you that law of dominance is not a universal law, which means it is not followed without any exception throughout the biology. It do have exceptions, and there are two exceptions that we are going to study: incomplete dominance and codominance. Correct?
Yes.
I ask you to go back and read upon incomplete dominance and codominance. Did you do that?
Yes.
Everyone? Yes? Okay. So having said that, having studied law of dominance, let's come to the exceptions of law of dominance. So if in a heterozygous condition, there are two different alleles present and one is dominant over the other, which means only one expresses fully, completely, and the other does not express at all, then it's called law of dominance. Now, incomplete dominance is where, let's start with the first exception, which is in complete dominance. And as it says, the dominance is there, but it is not complete, it is incomplete. So one allele is not able to express or exert its dominant totally and completely over the other allele in a heterozygous condition. Okay, so you can write down incomplete dominance. Write down. It's this incomplete dominance is where any one allele is not able to express itself totally. Any one allele is not able to express itself totally over the other allele in a heterozygous condition. Okay, so no single allele is completely dominant over the other one in a heterozygous condition. Now this means that because there is no complete dominance exerted by a single allele, then there has to be a blending of the trait due to both the alleles present. So write down, in the case of incomplete dominance, there is blending of traits that takes place. The blending of traits takes place. What do we mean by blending of traits, Atif?
Like a little bit of both the traits will be expressed.
Yes, so one trait will not be able to just skew the expression. It will not be able to mask the other. So the other one will also interfere with the trait. Now the possibility is that the dominant trait is not expressed totally, is expressed low or not expressed at all, due to which there is blending of the traits. Okay, right. So a little bit of uh, impureness or expression of both the traits is seen there. Now this is observed. An example is, there is a plant called snapdragon plant. So snapdragon's flower color. So this particular trait has incomplete dominance. When Mendel did the experiments to give to figure out to formulate these laws, basically he did not know of other plants and other genes and their behavior. He did only on peas and thought that every other plant behaved like peas, but that's not the case. Okay, snapdragon's flower color has two alleles for the gene. Now if it is capital R, then it is for red color. Okay, which is the dominant. And if it is small r, it is for the white color. Okay, now this one should be a dominant one and this one should be a recessive one, right? Normally following the law of dominance, one should be dominant and one should be recessive, correct?
Yes.
If the genotype is capital R, then what do you expect uh, the color of the flower to be?
Red color.
Yes. the flower is red. And if it is small r, what do you expect?
White.
Flower is white. Because both of these are homozygous, right? Both are the homozygous condition, correct?
Yes, sir.
But in a heterozygous condition, what should be the normal expression if law of dominance was followed? If law of
Sir?
if law of dominance was followed, what should have been the trait?
Red.
Red. Instead, in this case, the flowers are flower is pink. Now, what does this tell you? This capital R as an allele over small r as an allele is not completely dominant, right? There is an incomplete dominance, because of which there is a blending. So when you mix, if you have, like you all have done paintings in junior classes, you have mixed paints and colors. If you mix a red color with a white color, what will it produce? A pink color, right? So these colors are because of pigments present in the flower, correct? So the gene is giving rise to a protein, which is a pigment protein. Yes or no? This allele is producing a white pigment. This allele is producing a red pigment. But when present together, the flower shows blending in the pigment, and there is a pink color of the flower because both the pigments are blending with each other. So write down. Make this and write down. Snapdragon's flower color gene. Snapdragon's flower color gene shows incomplete dominance in a heterozygous condition. Shows incomplete dominance in a heterozygous condition. So just remember these three genotypes and their phenotypes. Now, because of this incomplete dominance, if I ask you, if I cross a pure line red with a pure line white, yes? This is a pure line red. And this is pure line white, correct? Everyone?
Yes.
Yes.
So if this is the parent generation, tell me what will be the progenies in the F1 generation?
All will be pink.
Sorry?
All the progenies will be pink.
Yes. So if you make the cross, you will realize that in F1 generation, all the progenies will have capital R and small r, correct?
Yes, sir.
This will be the genotype in F1 progenies, and all will be pink in color. Nothing like the parent, correct? No red, no white. If we go beyond F1 and we self-cross F1 to get F2 generation. Now tell me, what will be the genotypic ratio and phenotypic ratio in F2? Quickly.
Both will be 1:2:2.
Both will be?
1:2:1.
Yes. Otherwise, what should have been the ratio? So
Genotypic ratio should have been 1:2:1, and phenotypic ratio should have been 3:1.
yes. So if I make the cross like this, one progeny will be this, the other progeny will be this, and this, correct? Here, if you see the genotypic ratio, it is not affected, right? It is capital R is to capital R is to small r. That is 1 is to 2 is to 1, correct? But if we talk about the phenotypic ratio, now this will be red, right? Only this will be red and this too will be pink. So the phenotypic ratio will be red is to pink is to white, which is 1 is to 2 is to 1. You see that? So do you understand that incomplete dominance affects the phenotypic ratio due to blending of the genes, the blending of the characters, but it does not affect the genotypic ratio? Yes, it cannot affect the genotypic ratio, correct? Everyone, do you understand? Now write down, make this and write down. Incomplete dominance changes the phenotypic ratio but has no effect on genotypic ratio. And in the F2 generation, both is 1 is to 2 is to 1. Both are 1 is to 2 is to 1. Is this clear, everyone?
Sir, in incomplete dominance, will the genotypic ratio be always equal to the phenotypic ratio?
It depends if you are studying incomplete dominance with 1 gene inheritance or 2 gene inheritance. So saying that it will, in a 1 gene inheritance, it will always stay the same because you are talking about just 1 gene. In today, we will also talk about 2 gene interaction, dihybrid crosses. All these crosses are monohybrid crosses, right? We are only taking 1 gene with 2 alleles and we are making a hybrid of that. So if you are talking about monohybrid crosses, yes, every time in F2, the genotypic ratio will be equal to the phenotypic ratio in incomplete dominance, but might not be the case in dihybrid cross, in 2 gene interaction. Okay.
Okay.
Let me know when you're done drawing this. This is done, people. Shall we move forward? Am I audible, Atif?
Yes, sir. Done, sir.
Yes. Shall we go ahead? Done?
Yes, sir. I can.
Done.
Perfect. Okay. Hanifa and Hiba. Done, I guess. Okay. So this was the first exception to the law of dominance where it was not being followed. The second exception is known as codominance. Now, it is different from incomplete dominance in the manner where when 2 alleles here are present in a heterozygous condition, both are equally dominant, both express each other in the presence of each other. Now this is again a violation of code of the law of dominance, where because no single allele is dominant. Okay, both are dominant. So write down, in case of codominance, both alleles in a heterozygous condition express themselves equally, which means both are co-dominant, they are together dominant. The example for this is, you can understand this through the AB blood group. In humans, you know that humans follow ABO blood group system.
Yes, sir.
Yes. Now talking about ABO blood grouping, write down. In humans, there are, for the blood group, there are, for the same gene, there are 3 alleles. Okay. The first allele is A, allele for antigen A. It gives rise to a sugar which acts as an antigen on the surface of RBC. It gives rise to antigen A. Then there is allele B, which gives rise to antigen B. So there are, and then the third one is called small I allele. It's also called an empty allele because it produces no sugar. So if there is no sugar, there is no antigen. Is it clear? So it's like, if IA is present, there will be a sugar produced. If IB is present, there will be another sugar produced. They are slightly different, which makes them antigen A and antigen B. If small I allele is present, the third allele, it produces no sugar. So for blood group in human, A blood, there can be how many different types of blood groups can be there? 4, right? For blood group A, there are 2 possibilities. One is IA, right? The individual got the allele A for the gene from mother, and the same allele A from father as well. So this is a homozygous condition, and of course it will be A, right? The other possibility is IA and IO. In this case, there are 2 alleles. It is a heterozygous condition, but only A will be expressed, right? O will not produce any sugar. You understand? So,
Sir, IO is the same as small I, right?
sorry.
IO is the same as small I, right?
Yes, these are the alleles. This is the allele, and this is the genetic, the makeup, like the genotype I am writing. IO is this small I allele which produced no sugar. We write it as O. O means 0, nil, no sugar. That's what O group is. So for A blood group, these are the 2 possible genotypes. This is homozygous, and in this case, IA-IO, there is a law of dominance being followed because A is dominant over O. O is not expressed at all. Okay, so only A is expressed. So here are 2 alleles. In the presence of A, I does not expresses, only A expresses. So here it's normal. Law of dominance is followed, no codominance. The second group is B blood group. Can you tell me the genotype for B now? Atif?
IB or IBIO.
Yes, this is same homozygous, or if this is the condition, then again law of dominance is there, right? Third is O blood group. Can you tell me what will be the genotype for O blood group? O blood group is a
IO.
yes, it's a recessive group. So only possibility is this: to make O blood group, you need to get IO genotype or small I allele from both the parents. Okay, because this condition will give rise to A and B. So there is only one possibility. There is no codominance in any of these three cases. But when you go to the AB blood group, you know there is a AB blood group. And why do we call it AB blood group? Because the genotype here is IA, IB. And normally, following the law of dominance, it is a heterozygous condition, just like these two. So there should be one that should be expressed, the other should be not expressed. If it is incomplete dominance, there should be a blending and something between A and B must be there. But in this case, there is no blending, also no dominance. There is codominance. Both the sugars are produced and are present like, uh, like uh, and are present on the surface of the RBC, membrane of the RBC. So you will see both A antigen and B antigen, nothing in between, no hybrid thing. Is it clear? So this case, write down, in AB blood group, the alleles IA and IB are codominant. Both antigens are present together. Is it clear, everyone?
Yes.
Please note it down. Ask me if there is any doubt. Done? Done, everyone?
Done.
Others? Okay. So both incomplete dominance and codominance is clear to you?
Yes.
Yes. For incomplete dominance, there is one more example that I want you to read by yourself. Write down. It is the example for production of starch granules in pea plant. You know, but production of starch granules was not studied by Mendel because for that you need microscopy, like advanced microscopy, where you can see the size of these granules, starch granules. Okay. So that was not done by Mendel. Otherwise, Mendel would have realized that there is incomplete dominance in some cases. Okay. So write down as a homework for incomplete dominance, you have to read gene for starch granule formation in pea plant. It's there in NCRT. Okay. Will you read this? Everyone, Rifat, Atif, this is a homework. Okay.
Okay, sir.
It's the same thing as the Snapdragon plant. Okay, now moving from law of dominance. So this was all about law of dominance, what it says and what are the exceptions. Moving forward, coming to the second law of Mendel, which is known as law of segregation. Law of segregation, or it's also known as law of segregation of gametes. Also commonly known as law of purity of gametes. Law of purity of gametes. Write down what does this law tells. Write down, this law states that no that that do not show alleles do not show any blending while making gametes and each gamete receives one allele which is segregated randomly and independently of each other, which is segregated randomly and independently of each other. Is it clear, everyone? Written? So it means that gametes are always pure and receives only one allele. Receives only one allele. And this law, write down, law of segregation is the only universally accepted law. with no exception. Universally accepted law with no exceptions. And what do we mean by this? All through this time, we have been following law of segregation when we were making gametes, remember? So for example, even if there is a hybrid, a plant which is hybrid tall, right? So this character is hybrid tall, but it is tall, yes? In case of a pea. In case of a snapdragon, this shows blending. No dominance, incomplete dominance, and it is pink, right? And in blood group, A and B shows codominancy, right? All these three different things, whether they follow law of dominance or they do not follow law of dominance, but they always follow law of segregation. Because taking the example of this one, when you make gametes for the cross, remember that they always segregate independent of each other, right? The gametes are always pure, right? You understand? No matter when they were present together, whether they were blending or not blending, they were codominant, incomplete dominant, or dominant. When a gamete is formed, all these, they segregate independent of each other. This capital R will go independent of small r, and only one will be received by one gamete. Okay? And no blending will be there. Because next time, let's say, let me take an example of this one. Let's say capital R and small r, a plant is making gametes. So it will make either this or this as a gamete, right? When it was at this stage, it was forming pink flowers. Correct? Everyone understanding, Atif?
Yes, sir.
Yes, but when it made two gametes, now if this gamete goes and fuses with another R, should not show it like this. Let me show it like this. If this is crossed with another R, and as a result there is a capital R, what will be the color again? Red. But this came from a pink flower, you understand? And later on can form red. Similarly, this also, this recessive gamete which came from a pink flower can later on in the next generation again form white flower, which means that at the level of gametes there is no blending. The alleles come pure, they become pure. So gametes are always pure, they are never impure or blended or touched. Is it clear?
Yes, sir.
So this is law of segregation with no exception. Make this, then we'll move forward. Now the third law talks about, third law is known as law of independent assortment. Okay, and this law talks about the characters, two different characters. So to understand that law, we have to now move from a monohybrid cross, which means a single gene inheritance, to a double gene inheritance, or known as two gene inheritance. This means that now we will study two genes being inherited together, because in biology it's never a single gene that's inherited like alone. There are always many genes that are inherited together, right? So two gene inheritance, and for that we have to make dihybrid cross to understand. Is it clear, everyone? Now two gene inheritance means, let's take any two genes. Tell me any two genes from pea plant. Atif, there is a parent A and a parent B. Tell me any two genes that you want to study. Atif, Hiba.
Stem height and color of flower.
Yes, so let's say stem height, which means the height, which means you are saying tall. Let's say a tall, pure tall. Let's start with pure, okay? Pure tall with color of flower. You said color of flower, Atif?
Yes, sir.
Yes, so which color is dominant in flower?
Violet color.
Yes. Let's say pure tall with violet flowers. Okay, and the parent B will be pure dwarf, and because dwarf can only be pure, so no need to write pure. You can write dwarf with white flower. Okay. Can you also tell me the genotype for parent A, Atif?
Capital T, small t.
Capital T, capital T. It's pure tall.
Sorry, capital T.
Yes.
Then
Let's say capital V. Okay. For violet. Basically, you should take it for W because did I tell you that we take the alphabet from the recessive gene. That is the right notation, but NCERT, it does not follow the right notation all the time. To make you understand, for tall it takes capital T, and for dwarf it takes small t. So I'm taking the alphabet which correlates to the dominant genes. They both are dominant, and we are crossing it with what is the genotype of parent B, Atif?
small t, small v, small.
Yes, and small v. Okay, so when we will cross this, so this is the parent generation. First you have to figure out for F1 how many gametes are possible in this case. Gametes that this plant can make. How many different types of gamete it can make? It can only make one type of gamete, which is capital T and capital V. Because in gamete, one allele from each character goes, right? Yes?
Yes, sir.
So you understand that it will be capital T and capital V only, one type of gamete. And for this plant, it will be small t and small v only. And if we cross both of them, the F1 will be capital T, small t, capital V, small v. Correct? This will be the genotype for all. There will be no ratio. Does it make sense, everyone?
Yes.
And what will be the phenotype? The plant will be tall and violet. In this case, you can say hybrid tall and hybrid violet. Yes? Make sense?
Yes.
Now, this you have to do. If we self-cross F1, what will be the genotypic ratio and phenotypic ratio in F2? Can you figure out?
Yes.
It will take 2-3 minutes. It's going to be a long cross because follow the same rule. Find the genotype of the F1, which you have to cross, which is already given here. So basically, the cross that I'm asking you to make is capital T, small t, capital V, small v into capital T, small t, capital V, small v. This is the cross that you have to make. So figure out first the gametes, Then make the Punnett square and do the cross. Everyone, are you doing it? Rifat, Hiba?
Yes.
Yes, please. And let me know when you're done. Just say done. Okay, so did everyone successfully made the gametes?
Yes.
Yes. So tell me, what will be the possible gametes for this one? How many gametes it can form?
Capital T, capital V.
So one gamete can be capital T, capital V,
Then capital T, small v.
capital T, small v, right?
Then small t, capital V.
Yes, small t, capital V,
Then small t, small v.
small t, small v, right? There are four possible gametes, then this combination can form. So similarly, there'll be four for the other one as well because both are same. Capital T, small t, capital V, sorry. Capital T, capital V, capital T, small v, small t, capital V, and small t, small v, correct? So you see that a dihybrid cross is not four, it does not have four progenies, it has 16 combinations. So let's write down what will be the first progeny. Capital T, capital T, capital V, correct?
Yes.
This will be capital T, capital T, capital T, capital V, small v, capital T, small t, capital V, capital V, capital T, small d, capital V, small v, capital T, T, capital V, small v, am I doing it correct? Please keep cross checking. Everyone. Is everything correct till now? Yes, this one. Small t, small v. Is this correct, everyone?
Yes.
Yep. Now, looking at the cross, let's first figure out the genotypic ratio and the phenotypic ratio. Now, you have to tell me what are the different types, how many different kinds of genotypes are possible. First, I have to identify that. This is the first type, right? Can you see any genotype like this anywhere else?
No.
So this one is unique, right?
Yes.
Phenotypic ratio is 1:2. Now look at this one here. Can you see these two are same?
Yes, sir.
They are falling on the diagonal. These two are same. And there is no other genotype like this anywhere else. So 1:2, 2:2. Can you see this one here is unique? Capital T, small v, small v. Yes. And then these two, this one and this one, are same. Yes.
Yes.
So 2. Then this whole diagonal is same. All these 4 ones are same. Can you see that?
Yes.
Then again, these 2 are same. This is unique. These 2 are same. And this is unique. This will be the genotypic ratio in F2 of a dihybrid cross. Okay, where there is, it's following the law of dominance, and there is no incomplete or codominance or anything like that. So we see it's 1:2, 1:2, 4, 2:1, 2:1. Yes, Hiba, do you have a question? Hiba, any question? You unmuted yourself. No. Okay, so the genotypic ratio is clear, everyone?
Yes.
Now tell me the
So is there like a standard way to write the ratios, or can we write them in any order, like in like the exams and
like, see, technically you write any order, it's correct, right? Because the one way is to, whatever genotype you are writing, you can just mark that genotype here. So for example, I can start, easy is to go from one side and scan towards the end. So you will find this ratio 1:2, 1:2. You can also write 1:1, then this 2 and 2 together, 1:1, 2:2. You understand? But then you have to mention. If you go by this standard, then you do not have to mention. It's clear that you are following the genotypes on a diagonal, because see, it increases 1 2 1 2 4 2 1 2 1 and then decreases, right? It's same. You read from any side. You read from this side or this side, it's the same. Do you see it? Atif?
Yes, sir.
Yes. So you will not be technically, you will not be incorrect technically, even if you write in any other order, but then you have to mention which genotype you are writing about. Okay. Now tell me phenotypic ratio. Rifat,
Yes, sir.
how many different phenotypes can be possible? If we are crossing these two, what were the parents' phenotype? This was hybrid tall and hybrid violet, both, right? So how many different phenotypes are possible?
4.
Tall and violet, tall and white, dwarf and violet, and dwarf and white, right? If there are 4 alleles, then if there are 2 genes with 4 alleles, this could be the only possibility. Yes or no?
Yes.
So what will be the phenotypic ratio? How many of them will be tall and violet? Let's count. This will be tall and violet, right? 1. This will be tall and violet. 2, 3, 4. Then 5. This will not be tall and violet, right? This will be tall and white. 6. This will be tall and violet. 7, 8, and 9, right? Anyone else which is tall and violet? No. So 9 will be tall and violet. Now let's come to tall and what was the other one? What was the other parent?
White.
Hybrid, sorry, tall and white, right? How many will be tall and white? This will be tall and white. You see here, 1. This will be tall and white, 2. And this will be tall and white, 3. Yes? No other will be tall and white. So 9 is to 3. Now dwarf and violet. I find 1 here, dwarf and violet, 2 dwarf and violet, and 3 dwarf and violet. Yes? Is to 3. And only 1 will be dwarf and white. Pure recessive for both. You understand? Is it clear, the genotypic ratio and the phenotypic ratio, people?
Yes.
This is the genotypic and phenotypic ratio in the F2 generation of a dihybrid cross. Now by this cross, what did you learn? What can you tell me? We started with a hybrid tall and hybrid violet. Tall and violet, tall and violet. Was there any parent which was tall and white, or dwarf and violet, or dwarf or white to begin with? No, right? If we talk about the genes, look here. Tall was in both the parents, tall was present with violet only, right?
Yes.
Yes or no? So through dihybrid clause, law of independent assortment was given. Now write down what is law of third law. Write down law of independent assortment. Write down this law states that This law states that the alleles of two or more different genes alleles of two or more different genes gets sorted into gametes independently of each other. It simply, in other terms, means write down. In other words, it means the segregation of one gene, the segregation of allele of one gene, the segregation of allele of one gene does not influence the segregation of allele of another gene. The segregation of allele of one gene does not influence the segregation of allele of another gene. Is it clear? And this is visible here. See, this gene for height had two alleles, right? One was capital T and the other is small t. And this gene for flower color also had two alleles, right? Capital V and small v. Make sense, everyone?
Yes, sir.
But during the gamete formation, you can see that every allele of every gene made a combination with every other allele of every other gene. Yes or no?
Can you repeat?
Every allele of every gene, capital T or small t, every allele of each gene, or I can say each allele of each gene made a pairing with each allele of each other gene. Yes, that's why there were 4 gamete combinations possible. So that capital T gets the turn to come with capital V and small v, and small t gets a turn to come with capital V and small v, right? So this means that one gene was not influencing the other gene. Both the genes, though they were present together, they were also being inherited together, but the genes stayed independent of each other. When the time to make gametes came, both the genes behaved independently, right? This is what is law of independent assortment of the genes. Okay. Law of independent assortment is at the level of gene, because there are multiple genes present now in a dihybrid cross. But law of segregation was at the level of alleles in one given gene, in a single gene. Is it clear? The difference between, what is the difference between law of segregation and law of independent assortment? Is it clear? Riba, Rifat? Not speaking. Yes? Okay. Good. So, law of independent assortment, what do you think? Is it a universal law? Oh,
Yes.
no, it is not a universal law. It has exceptions because it talks about two genes and their behavior with each other. So when Mendel did all these experiments in P, he was not aware of the fact that genes can either be linked. There are many genes which are linked together. Okay. So whenever one gene is inherited, the other genes comes with it. So this was not known at the time of the pea plant experiment of Mendel. So write down, law of independent assortment is not a universal law. Not a universal law, but law of segregation of gamete is a universal law. So there is no exception in the fact that alleles segregate independent of each other to make gametes in a given gene. That holds true all the time. But this is not true all the time, that genes segregate independent of each other all the time. Do you understand this? It was doing that in case of pea plant. So this is the normal scenario where they segregate independent of each other. All the alleles of all the genes segregate independent of each other. But it has exceptions. Write down. Law of independent assortment has exceptions in the form of linkage of genes. In the form of linkage of genes. Is it clear, everyone?
Yes, sir.
Yes. So to understand law of independent assortment, let's do a question first. Write down the question and you will solve and tell me what is the genotypic and phenotypic ratio. It is a dihybrid cross and you have to follow law of independent assortment. Write down. A heterozygous round seed, a heterozygous round seeded plant. Let's not write seeded, just write a heterozygous round and homozygous tall. A heterozygous round and homozygous tall plant was crossed with wrinkled and short plant. Heterozygous round and homozygous tall plant was crossed with wrinkled and short plant. Okay.
Okay, sir.
If this is a parent generation, tell me in the F1 generation, what will be the genotypic and phenotypic ratio? Do it and tell me. Let me know when you're done, okay? I'll be back. I'll just get some water. Okay, Atif, Rifat? You know the steps, right?
Yes, sir.
Okay. Anyone who has done? Rifat, will you please help me? Like, I'll follow your lead. Tell me the genotype for the parents. What will be for this parent A? The genotype.
It will be capital R, small r,
Capital R, small r, and
capital T, capital T.
capital T, capital T. Correct. And for this parent B,
It will be capital R,
it's wrinkled.
yes,
If it is wrinkled, it cannot have.
small r, small r, small r,
Okay.
and small t, small.
Yes. Correct. So let's first come to gametes. Same method. How many gametes can be formed by this? How many different types of gamete? Tell me, Rifat. Tell me the gametes for parent A.
Capital R,
Hmm?
capital T, no, capital R,
Sorry, be a little loud, please.
capital T,
Capital T, just 1 minute. So capital R, capital T is one gamete, and
small r, or capital.
small r, capital T is another gamete, and are there other gametes possible?
Mhm.
No, right?
Yes.
In this case, how many gametes are possible?
Small r, small t.
Small r, small t, and that's it.
Yes.
Yeah. So if you cross, if you make a cross, what will you do? Trick question. You just have to make, because one gamete is capital R, capital T, small t. Here it's small r, small t. The other gamete will also be small r, small t, just for the sake of completing the square, right?
Yes.
Now tell me, this progeny will be capital R, capital T, small t. This will also be capital R, capital T, small t. This will be small r, capital T, small t. And this will be small r, capital T, small t. Am I correct?
Yes.
Everyone has done this only. So what is the genotypic ratio? These two are same, right? Yes or no? And these two are same. So it's 2:2, correct? Yes or no?
Yes.
And what is the phenotypic ratio?
Twisted.
Round, tall, and then wrinkle, tall, right?
Yes.
So 2:2. Correct. So we started with a round and tall and wrinkled and short plant, but in the first generation, we see that there is no short. Either it is round tall or it is wrinkled tall. Yes,
Yes.
everyone. Now the interesting bit. Uh, if I take this plant and self cross it from the F1, I'm taking capital R, capital T, small t, and I am self crossing it. with capital R, small r, capital T, small t. Now, can you tell me what will be the genotypic and phenotypic ratio in the next generation? This time you have to give me complete answer. Rifat, Atif and Fatima, Hiba, Rifat, Atif and Hiba. Everyone, was I audible?
Yes, sir.
Do this and tell me what will be the GR and PR in F2. And try to be quick now.
Phenotypic ratio will be 9:3:3:1.
Okay,
And genotypic ratio will be 1:2:1:2 4:2:2:1:1.
and how did you do that? Because it's same like the last cross, right?
Yes, sir.
So you just have to remember, people, that if it is heterozygous for both the alleles, can you see both the parents are heterozygous for both the genes, right? Hybrid. This is hybrid red, hybrid tall, hybrid red, hybrid tall. If both are hybrid, then your genotypic ratio is going to be 1:2:1:4:2:1:2:1. Yes. When there is no incomplete dominance or something given, the phenotypic ratio will be 9:3:3:1. Yes.
Yes, sir.
Now, if I just twist this question to make it difficult, I'm telling you that in this plant, look here, write down the question and keep it as a question in your notebook. It's a very important question. If a hybrid round, which is this hybrid round, and pure tall is crossed with another hybrid round and a medium plant. So which, when I say medium, it means that there is an incomplete dominance here, right? You understand? Hybrid round and tall.
Sir, can you repeat the question?
Hybrid round and tall plant. Hybrid round and pure tall plant is crossed with hybrid round and a medium plant following law of incomplete dominance, which means in this allele here, I am telling you, just assume that there is incomplete dominance. Then only it can be a medium plant, no? You understand,
Yes.
people? If there was pure dominance, then it would be a tall plant, a hybrid tall plant. Yes, because of incomplete dominance, it will be a medium plant, and only this will be a short plant. You understand? Short, medium, and tall. If this is given, then there is an incomplete dominance. You understand? Do you understand?
Yes, sir.
Yes. So question is written. Hybrid round with pure tall is crossed with hybrid round and a medium incomplete dominance, medium plant. Now give me the genotypic and phenotypic ratio in the next generation. Give me, find the genotypic ratio and phenotypic ratio considering the incomplete dominance between among the alleles of the gene of height or the gene for height. Question written? Everyone, question is written?
So,
Now, please do it and tell me. Hiba, are you there? Can you speak? Am I audible to you? I could not hear if you are speaking anything. I saw that you unmuted yourself, but you are not audible to me.
Yes, sir.
Okay, yes, now you're audible very feebly. Please do it and tell me the answer. So you have to write, you have to give me the answer in the chat. Write the genotypic ratio and phenotypic ratio. You can use comma or just cap if you cannot put semicolon.
sir, how will you find the Punnett square for this?
How will we find?
The Punnett square for this.
First, you have to find the gametes.
Yes, sir, but I got like 2 gametes for one plant and 4 gametes for the other plant. Like, how will we?
Yes, so you just have to make 2 on one side, 4 on one side, make a cross. There will be 8 progenies.
Okay.
Understand? Or what you can do is what we did here, Atif. If you want to make it equal, remember what I did here? There was only one gamete, right? I wrote twice. So if there is only 2 gametes, then they will be the 2 more gametes, right? You understand?
Yes, sir.
Yeah, you can do that. So do that. So make a 16 chambered square, everyone. And then tell me the ratio. Please tell me when you are done. Okay, Atif, Rifat, and Iba. Okay, I've received one answer. So let's, Atif, will you lead me? Let's solve this.
I'm doing the question.
Okay, you're doing it still. Please finish
Yes.
and finish fast. Quick. Try to be faster now because I make you practice these questions in front of me. If you have any doubt, you can ask me. And if you are doing fast and you make mistakes, we can figure it out. So you have to practice it yourself at home.
Done.
Okay, done. Okay, so let's begin. Atif, tell me. First, we know the genotype for the parents. So tell me what are the gametes that are possible for parents A and B. For A first, tell me.
Capital or capital T and small or capital T.
Okay. And for parent B, what for B?
Capital or capital T, capital or small t,
Okay.
small or capital T, and small or small t.
Okay. Right. be correct. Correct, everyone?
Yes.
Make it 16. Now tell me. Solve all this. Just keep cross-checking. If I do something wrong, tell me. Am I correct? I'm doing it correct?
Yes, sir.
Yes. Now tell me, what is the genotypic ratio? Atif. What is the genotypic ratio?
Sir, I got 2:4:4:2:4. I don't know if that's correct.
You got 2:4. Which is 2? So let's, so this genotype,
Capital R, capital R, capital T, capital T. There is two
2.
of them.
Yes, perfect, right? 2:2. Then these two are done. Then which is the another 2?
No, sir. The capital R, the first and third one, not the second one.
Yeah, yeah. This moment. Yeah, okay. So this one and this one, right?
Yes, sir.
2:2. Then what is the other 2? This one and this one?
So that one, four of them is there in the
Yes.
first or third one.
1, 2, then 3. Where is the fourth one?
Fourth in the first row, the third one.
This one.
Sorry, the first row, third column.
First row, third column. You're talking about this one, right?
No, sir. Yeah,
This one. This
so
one.
yeah, that one.
Yeah. So 2:4.
Yes, sir.
This 2. What are the other? I think these are 2, right?
Yes, sir.
This 2. This is 1, 2, 3, and 4, right?
Yes.
2:4 is to 2:4 is to 2, right? Is to 2.
Yes, sir.
If you're writing it like this, then you have to write the genotypes on top of each. Then it will be correct, not wrong. Otherwise, you arrange it 2:2:4 4:2:2. Okay.
Yes, sir.
So if I arrange it properly, it will be 2:2:4:4. So do you see now that the genotypic ratio has changed just by changing the parents that we begin with? So we started with this parent. So the genotypic ratio has changed, right?
Yes, sir.
What about the phenotypic ratio? What are the possibilities? This was round and tall, but for phenotype, you have to consider medium plant also now. So what are the different phenotypes possible? First tell me. Either a round and tall is possible. Round and medium is possible. Yes.
Yes, sir.
And what else?
Wrinkled and tall.
Round and short, is it possible? Round and short?
No, sir. Short is not.
Yes. Then wrinkled and tall is possible, right?
Yes.
Is two. Wrinkled and medium is possible.
Yes.
Wrinkled and short is possible.
No.
There is no wrinkled short. So there are these four phenotypes possible. Now tell me the ratio. How many are round and tall? 1, 2, 3, 4, 5, 6. Yes. Is it clear? Round and tall are 6. Yes, everyone. Round and medium 1, because now capital T small t will be medium, right? 1 medium, round and medium. 2, 3, 4, 5, 6. Correct?
Yes, sir.
Wrinkled and tall. 1 and 2. Yes.
Yes, sir. And then wrinkled and medium is.
This is also 2. 1 and 2. So in total, there are 16 progenies. But you see that the ratios are different for F2 now because it depends on two factors. The phenotypic ratio depends on incomplete dominance that we have considered, and the genotypic ratio depends on the parents that we started with. Okay? Because now there were different gametes. Is it clear to all of you, everyone?
Yes, sir.
Everyone has done the same cross and written the same? Anyone whose answer was different?
Sir, my genotypic ratio was wrong. I corrected it.
Yes, but you did not see the genotype properly, but you solved it like this, right? All your genotypes were this only.
Yes, sir.
You could not like just count each genotype, right? The one way is when you make it, one way is keep a pencil with you, and when you see one genotype, just make a cross that it's counted. You understand? When you see that genotype again, just make a cross. Is it clear?
Yes.
For your boards, it's a tip. Then you can remove that after you are done writing. Okay, cool. So if this kind of question comes, will you be able to do it? Now, do you now understand, Atif, your question which you asked me up in the class, that if due to incomplete dominance, will the genotypic and phenotypic ratio always the same? And I said it will not be. Yes?
Yes.
Yeah. In a dihybrid cross, it's different. Now, the last thing that we'll do today is in all these laws that we studied, law of dominance, law of segregation of gametes, and law of independent assortment. All these laws they came from Mendel's work, but when Mendel did the work and when Mendel actually made all that experimental observation. He noted down all the observation. He wrote down the results and he talked about the genetics that he did. He did not write as laws. He did not say there are 3 laws. He just wrote everything. This was the result and this was not the result. Yes, Hiba, do you have a question? Hiba, you're not audible if you're asking something. Can you please write in the chat if you're asking a question? I'll take it from there. So Mendel's work was not appreciated till the very late, till the beginning of the 20th century, like till the 1900s. Mendel's work was not appreciated and it did not reach popularity. It did not reach the masses. Okay. And why it did not reach the masses? There's a question. This is a question which can come in board. So write down the question. Why did Mendel's work remained unrecognized? Yes, Hiba, are you saying something? You have rejoined to audio. Can you speak? Why did Mendel's work remain unrecognized till the 1900? Remained unrecognized till the 1900. Now there are 3 reasons behind it. Okay. The first reason was that there was the communication at that time, especially the scientific communication at that time, was very, very limited. It was not the age of internet that any scientist who published something, the next day the world knows about it, anyone can read it. So the work remained unrecognized because it could not reach a lot of geneticists around the world, a lot of biologists. So not proper to say geneticist because he started the field of genetics. So Mendel, is known as father of genetics. Everyone, Atif? Gregor Mendel.
Sorry, sir.
Gregor Mendel. Mendel is known as father of genetics. You know that?
Yes, sir.
Yeah. So at that time, other biologists, other contemporary scientists, the work could not reach them, okay, as fast as it should have. So write down the first reason was the communication was not easy. Communication was not easy. And the work could not reach wide audience or could not be widely publicized. Communication was not easy. It is research work, could not be widely publicized. Second reason was Mendel, for the first time, because Mendel did not know about genes. Okay, is it clear? So when Mendel did all this thing, he did not use the word gene, but he used the word factors. He called all these things which are being inherited as factors. So he said that for every character, there are factors in our body which are passed on from one generation to another through the gametes. And these factors are discrete. They do not blend or they do not form combinations to give rise to other kind of factors. So write down, he said, Mendel, second reason is Mendel's concept of genes, in bracket write down, He called them factors. Mendel's concept of genes was was a, was stable and discrete units. Stable and discrete unit that contains a pair of alleles which never blend with each other. This concept was not accepted. This concept was not accepted by other scientists of the time. Is it clear? Is it clear, everyone?
Yes, sir.
Yes.
Now, why it was not accepted? Because scientists found it difficult to realize that, okay, if they are not blending, though it was true because Mendel showed it through his experiments, it was true, but he did it just in peas. And scientists argued that in humans, do you think in pea plant there is a tall and a short, right? Yes or no?
Yes, sir.
But what about humans? Do you see in humans there is a tall and a short only? A fixed tall height of 6 foot and a fixed short height of 4 foot? Is it like that? No,
No.
right? We have a range, we have a whole gradation, and height could be anything in humans. We see all heights in humans. Every foot and every inch is there in human as a height, right? It goes till a higher limit, like there is a tallest person which is present on the planet, and there is a shortest person, but there is a range in between. Yes or no? So they said that for genes like height for human and for genes like skin color, in skin color also, it's not like flower color. Like humans are not either black or white. There's a tone, right? There is a gradation of skin color. Yes or no?
Yes.
So scientists did not accepted this uh, uh, for everything. They thought that Mendel has missed something. So that was the second reason. And the third reason was Mendel was the first person, for the first time, he used the concepts of mathematics to prove or to explain the phenomena of biology. So write down. Mendel's approach of using mathematics to explain biological phenomena. Mendel's approach of using mathematics to explain biological phenomena was new and unacceptable to many scientists of that time. Was new and unpopular or unaccepted. You know who was Mendel? Who was Gregor Johann Mendel? He was a physicist. His education was in physics, and by profession he was a priest. Okay, and he did use mathematics to explain biological genetics. So that's why he's one of like my favorite characters in science, in history of science. So do you understand why the work remained unrecognized till 1900s? Yes. Now quickly I will also tell you that there were 3 scientists, and these 3 scientists were Hugo de Vries, von Schermack, and Carl Correns. These 3 scientists independently, write down, they independently discovered Mendel's work much after he has given it, okay, and he's gone. They rediscovered Mendel's work and gave the work the status of laws. Status of laws. So these laws that you are studying, law of dominance, law of segregation, law of independent assortment, these laws were not exactly given by Mendel. It was when independently this work was later found out, and specifically Hugo de Vries is the most important person who raised the work to the status of laws. So who gave Mendel's laws? If this is the question, and if Mendel is not in the option, don't get confused, because Mendel did the work. He wrote it as a book, as a novel, like everything mathematical and everything is written in it, results and all those things. But to make it laws, to make 3 laws out of it, it was mostly Hugo de Vries and Schermack and Correns. Is it clear, people?
So,
Yes, so just remember this. And there is one more thing. So write down the homework before you come to next class. You have to study 3 things and come. First thing that you have to study is you have to revise meiosis. So I'm not giving it again as a homework. Meiosis will come multiple times. So there is chromosomal theory of inheritance. Chromosomal theory. So you have to read chromosomal theory, what it is. Of inheritance, we'll do this, and then we'll come to linkage and recombination. Linkage and recombination is an exception to the 3rd law of Mendel. Okay. Is it clear? Written homework. Any doubts? Anyone wants to ask something? If not, I'll let's declare the class here. Please go revise whatever we have studied and come prepared in the next class. Okay, everyone? Okay, then